Hello, everyone. My name is Katie Bogue, and I'm a third-year biochemistry undergrad at UCLA. My faculty mentors are Professor Margot Quinlan from the UCLA Chemistry and Biochemistry Department and Professor June Allard from the UCI Mathematics Department. And today I'm going to be sharing my research on a project focused on computational modeling of FH1 domains. So the overarching goal of my project is to create a robust model of FH1-mediated actin polymerization that provides structural explanations and ideally predictions for the effects of N-terminal dimerization. This will hopefully make more sense as I continue, but please keep this overall goal in mind as I go into some background. So to start, I want to explain a little bit about what actin is. Actin is a protein component of the cytoskeleton involved in many structural components, from microvilli to stress fibers. In addition to being involved in many different structures, actin itself can form many different structures. You can have long linear filaments, you can have branched filaments, you can have bundled actin, etc., etc. But what makes actin particularly unique and important is its role in cytoskeleton dynamics and cell motility, and this is due to actin's ability to polymerize and depolymerize. This allows for motion as actin monomers are being added to one end and taken away from another end. A vast number of actin-binding proteins enable actin to form different structures and play different roles. One such actin-binding protein is the formin, which is the focus of my project. Formins accelerate the formation of new actin polymers from actin monomers, or nucleation, and they control the polymer growth rate, or polymerization rate. Formins create actin filaments, so they elongate actin linearly, and they're composed of the formin homology, or FH1 and FH2 domains. The FH2 domain is this donut-shaped homodimer that is primarily involved in nucleation, but it encircles the growing actin filament during elongation. But what my project is focused on are the FH1 domains, which allow for much faster polymerization in the presence of profilin. So profilin is an actin-binding protein that can bind to these proline-rich motifs, or PRMs, along the FH1 domain. And the FH1 domain is essentially this long, I sort of think of it as tentacle-like domain that comes off of the FH2 domain. And the PRMs along this tentacle are able to bind to the profilin actin and then swing up and deliver the actin monomer to the growing actin filament. As you may have already been able to ascertain, but I'm now going to explicitly state, formins are dimers. So formins are joined at the FH2 domain, seen here in orange, but they're also typically joined at the N-terminal dimerization domain, noted here as the pink DD domain. However, a lot of research is done with C-terminal constructs that don't actually have the dimerization domain, the N-terminal dimerization domain present. And theoretically, that could have significant impacts on polymerization rates. In fact, preliminary work from the Quinlan lab suggests just that. So shown here are results from experiments using FOD-B, which is a formin. On the y-axis here is elongation rate, and we can see that when we use a C-terminal construct, elongation is noticeably faster than when we use a construct that includes the N-terminal dimerization domain. I also want to briefly touch on disordered proteins. So many proteins contain intrinsically disordered regions, or IDRs, that lack a formal structure. And FH1 domains are generally considered IDRs. And for a long time, based on the structure determines function paradigm that you're taught in, high school biology, we used to think that IDRs didn't contribute much to protein function. However, after observing many proteins that rely on IDRs, including formins and their FH1 domains, we know that IDRs can be crucial to many functions, but we don't fully understand how. This is particularly relevant when we think about computational modeling projects, because it's much, much easier to model structure than it is to model lack of structure, to model disorder. And so our project, in seeking to answer questions about formins, is also seeking to answer the question, how do we computationally model disorder? How do we model intrinsically disordered regions? So today I'm going to talk about a specific problem, a specific improvement to the model that I have recently made, and what that improvement tells us. I'll begin with a little bit of an overview of the original state of the model. We start with the simplest model, a one-step model, where we use the first step of elongation, which I refer to here as capture, as a proxy for the overall polymerization rate, because literature suggests that this is the rate limiting step. And so this is the step where you go from a sort of a naked foreman, where you have the FH2 domain in pink here, and the long tentacle FH1 domain down here. And this first step, this capture step, is the step where profilin actin binds to the PRM. And in order to determine this capture rate, in order to model these FH1 domains, these disordered regions, we use what we refer to as occlusion probabilities. So we simulate a bunch of different possible conformations of the FH1 domain, and we determine the fraction of those in which the PRM is occluded, meaning that profilin actin cannot bind, as opposed to conformations where the PRM is fully accessible. And we use this occlusion probability to determine the overall capture or polymerization rate. However, we ran into an issue using this one-step model, and this was that literature suggests that there should be a decrease in polymerization rate with distance from the FH2 domain, but our model was not showing that, which I'll get to in a second. But here is a figure from a Quartermanche and Pollard paper that used BNI1 constructs with the same PRM sequence, this PPD, at different PRM locations, at different locations along the FH1 domain. And what you can see is that when the PRM is closer to the FH1 domain, this black line up here, the elongation rate is faster than when you're farther away from the FH2 domain. But when we use this initial one-step model, that is not what we see. So over here, I have a plot from our simulations. Each dot represents the polymerization rate, or again, just the capture rate here, of an individual PRM. The x-axis is the distance from that PRM to the FH2 domain. The y-axis is the log2 of the polymerization rate. And we would expect to see a trend kind of like this that I'm showing with my mouse, where it decreases as you get farther away from the FH2 domain. But very clearly, that is not what we see. We don't really see any discernible trend. So in order to see the expected trend, we made some improvements to the model. And the improvement that ended up having the most significant impact was adding in an additional step. So moving from a one-step model to a two-step model. And the step that we added in was what I refer to as the delivery step, where the bound profilin actin swings up and is delivered to the growing actin filament. And the addition of this step added in two additional rates, the rate of delivery as well as the reverse rate of capture. And we again model both of these rates, the capture rate and the delivery rate, using occlusion probabilities. In this case, the relevant occlusion probability for delivery is the occlusion at the site where the monomer is added to the growing filament. And after modifying the model to include this additional step, we do see the expected trend, where polymerization rates decrease as you get farther away from the FH2 domain. So what we can conclude from this is that while the first step, what I refer to as capture, may in fact be the rate limiting step, incorporation of an additional step, what I refer to as delivery, is necessary to explain the effects of PRM location on polymerization rate. And so just to put what I discussed today into context of the model overall, I discussed one particular improvement that we made to the model, going from one step to two step. This is part of the overall goal to develop a robust model of FH1 mediated actin polymerization. All of these individual changes add up to help support a model that we can feel confident in, moving on to the larger goal, the ultimate goal of providing structural explanations and predictions for the effects of N-terminal dimerization. In order to be confident in our predictions, our explanations, our understanding of N-terminal dimerization, we need to be confident first in the model. And all of these foreman-specific goals are also in service of a larger goal, which is to increase understanding of disordered regions and proteins. Because if we can develop a strong model to answer our question about N-terminal dimerization, we're also answering a question of how can we model disordered regions. All right, before I let you guys go, I just want to thank a couple people. Special thanks to Camilla Kraus, Carolyn Wu, and Margot Quinlan from the Quinlan Lab at UCLA. Robert Taylor, Laura Clemens, and June Allard from the Allard Lab at UCI. And I would also like to thank all the staff at the UCLA Undergraduate Research Week office for making it possible for me to share my work with you today. Thank you.